DNA replication, transcription, and translation. Information stored in DNA is used by the cell to pass on information to its offspring in the replication process. The information is also used in gene expression, the DNA to RNA to protein process. Proteins do most of the work in a cell. This process is called the central dogma of biology, meaning that all of life's activities depend on this information flow. Genes are segments of DNA that provide each cell's instructions for daily life. Each gene contains a code for the cell to make one protein or a few related proteins. The average gene is 12,000 base pairs long, but they can have as many as 300,000 base pairs. Genes are located on particular segments of chromosomes. The human genome has about 20,000 genes distributed on 23 chromosomes. Each chain is formed by strong covalent bonds between the sugars and phosphates together, forming a sugar-phosphate backbone. The nitrogenous bases on one chain bond to complementary bases on the other chain with weak hydrogen bonds, AT and CG. The strong bonds in the sugar-phosphate backbone create a strong molecule. The weak hydrogen bonds allow the DNA molecule to be split, copied, and then put together. The double helix is made up of two strands. In this drawing, the backbone of one strand is shown as orange and the other as blue. The bases in the orange strand are the complements to the bases in the blue strand. As a result, the orange and blue strands are complementary. That the two strands are complements of one another is critical to understanding how DNA can be copied with great accuracy. The order of the bases on the blue strand is complementary to that of the orange strand. The complementary pairs are A and T and C and G. DNA replication is a process that involves copying DNA to make two identical daughter DNA molecules based on one parental molecule. When organisms grow and reproduce, cells divide. Every new cell needs a complete copy of DNA, its own set of blueprints in the nucleus. DNA replication occurs every time a cell divides. We learned that enzymes speed up chemical reactions. They also have essential functions in DNA replication. Creating an identical copy of the DNA double helix first requires splitting the two complementary strands apart. An enzyme does that by moving along the DNA molecule and separating it into two strands, just like unzipping a zipper. Next, other enzymes help make a complement of each strand. They add a strand of complementary bases to each base on the strand from the original DNA molecule. The result is that each of the original strands generates a double-stranded DNA molecule. Two daughter DNA molecules have been made. Each daughter molecule is made of two strands, one strand from the parental DNA and one newly created strand. Can you see how the daughter DNA molecules are just like the parent? Don't worry that one looks upside down.